Oh, I have to acknowledge. All right, let me know when you're ready. You guys are all ready.
Yeah, we're ready.
All right. First, let me thank all of you for being here today. And the invitation to speak here virtually at Orange Coast College is, again, it's a great pleasure to do this. So I'm particularly thankful to Dr. Melissa Ferguson for her invitation. And I cannot believe it's been 20 years since we were in graduate school. So let me make sure I state that clearly, because my mind is completely blown by that idea. And so, all right. So let me give you a little background. I think what I'm going to do today is go over briefly what behavioral neuroscience is. And then what I'll do is talk a little bit about how I became a behavioral neuroscientist and some of the interesting work we do in our laboratory. So I'm going to give you like the full background of who I am a little bit. And so first start off just a little bit, my area of research in psychology is the behavioral neuroscience of fear. So before I get into my research in fear, I think it's important to define what behavioral neuroscience actually is. Okay. So how I like to define behavioral neuroscience is the study of how the brain contributes to psychological processes and behavior, right? And so there is information always coming into the brain, and there is information that is processed by the brain. And then that process information in some ways is important for then the outward behavior. So if we think about the brain in humans, right, it is a 3-pound mass of what we call neural tissue. Neural tissue are made up of many different types of cells. Most notably are neurons. And there are about 100 billion, for example, neurons in the human brain. And roughly about all of those different individual neurons potentially contribute to making 100 trillion connections. Okay, so when I put a picture here of the brain, it's a mass, but that mass is made up of so much. And what I'd like to do is sort of get at a little bit about these different functions of different brain regions as they relate to fear. So I'll walk you through that. So in my research, for example, fear is the psychological process that I study. And fear itself is very important to all of us, right? So think about psychological processes. When I say fear, and I'll define it at a later point, there are many different types, right? Fear is part of a series of emotions. We also have other psychological processes such as memory and motivation. Now, all of these things are invisible, right? You can't see memory. You can't see fear per se in some ways. And you can't see motivation directly, right? And so we have to infer their existence by carefully examining changes in behavior, okay? And so I'm showing you a human brain, right? And however, we can't open up the human brain just to study, right? And so we often in behavioral neuroscience, we focus on studying animals where we do have brain access, such as a rat or a mouse. Again, the overarching idea is that the same psychological processes, particularly for my research, which is fear, are shared, whether we're talking about humans or we're talking about a rat or a mouse. Okay. So, all right. So I wanted to orient you guys a little bit on where I actually am. I'm not just in this computer, but I am somewhere far, far away. In fact, my laboratory and where I teach is in Albany, New York, which is denoted by this purple dot. Really, really far away, I would say, from Orange Coast College, as far as you can get almost. We're in upstate New York, really close to the border of Vermont and Massachusetts. And again, I thought I know a little bit about the geography of California, given that I was born and raised there. So I think I'm pretty spot on, right? Right below, yeah, Orange County is right there. All right. And I know that you guys are in Costa Mesa. So how did I actually get all the way to Albany? And so how did I become a neuroscientist? And I think this for me, it was like a long journey. And in fact, probably the journey is not so different from where you guys are currently. So I'll get to that in a second. So I was actually born and raised in Southern California, particularly in and around the Los Angeles area. None of my parents actually went to college. In fact, my mom raised 3 children on her own, working 3 jobs at one time at one point in her life. And so it really sort of left an important impact on, working hard and being responsible. And so for me, it was easy that when I was in high school, I had to contribute. And so when I was in high school, I worked many different jobs. I worked at a food stand, chopped vegetables, I cooked, I cleaned movie theaters. I worked at a hardware store. So my path is not pretty straightforward. I even sold clothes from Ross, yes. So this is important, right? And I think for me, it was an important foundation in learning how to work hard, right? And the one thing is that the one aspect of my life at the time was that I really wasn't focused on high school, nor was I really interested in college at the time. And so I think part of that is, when you're raised in a family where priorities are not necessarily on education, it's more like having enough to have enough for your siblings and your family. So other things come into sort of priority, I would say. Well, after high school, I barely graduated high school. Again, like I said, it wasn't a priority. Given that my mom put a special emphasis that I would go to college. And so the long story is I grew up in Los Angeles. I grew up in some interesting areas of Los Angeles. And we moved a number of times. And so I finally graduated my last 2 years of high school in Burbank, California. And so my first year after I graduated, I went to Pierce College, which is a community college in the Valley region of LA, Los Angeles. And then I went to Glendale Community College for a semester. So one semester at Pierce College, another semester at Community College in Glendale. And then I ended up going to Pasadena City College. So at some point I had moved to Pasadena. And so it was an interesting period because for me, I saw it as an extension of high school because everybody I knew was, going to school in an area. And I really wasn't focused on my academics. I was really focused on trying, having a life, working, and going to school. And so something interesting happened in that process. To be honest, like I said, I wasn't doing particularly well as a student, grade-wise. In fact, at Pasadena City College, I ended up actually being on academic probation while working a full-time job. So during my second year at Pasadena City College, something finally clicked. And what clicked for me was finding that I had an interest in psychology. In fact, the two courses that I took, and most memorably, was a course in Introduction to Psychology, and the other course was a Research Methods in Psychology course. And I really became excited and intrigued by the idea that you could design an experiment to answer a question about how we behave, right? And so that blew my mind. I mean, the Research Methods course was like, oh, wow, this is cool. This is how experiments are done. Now, if I understand how these pieces work, I can design my own experiment. And in fact, I did. And it was looking at the relationship of success for community college students and the impact of having self-confidence, self-efficacy, and how these are important for student success. And so that really got me excited. And I learned to study for the first time, and I put effort in. So I actually had a goal, you know. I may not have been the smartest student, but I was a student that I had a plan, and I was willing really to work hard. So during this time, eventually I got my associate's degree. My grades went up, you know, because like I said, I worked really hard. I was sort of on this mission. And I was thinking about transferring. And so I thought about, you know, going to a UC or a CSU. And so, and you know, I thought about it a little bit, and I didn't think I would qualify for certain schools, given realistically my grades. Even though they were improving, it was sort of, you know, one of those things when your GPA takes a hit, it takes some time to get it up. And so I transferred to Cal State Long Beach because from what I've read and what I heard from other professors that I had, it had a very good psychology department. Also importantly, it was not too far from the beach. And so I really took my own initiative. While I know there were, you know, you can have academic advising, I read the university graduation, you know, bulletin and everything. And I figured out my course of action, how I had to finish my courses in 2 years, you know. So I was on this mission to excel and finish. So in that process of taking psychology courses, so over here is the psychology building at Cal State Long Beach. I took my first neuroscience course class. And let me say, from maybe after the second week of being in the course, I had this idea that I would want to teach this exact same course someday, right? So that was really pivotal in me becoming a neuroscientist. So I had visions of being a psychologist, you know, in the more traditional sense, but I didn't realize the depth in which psychology includes the ability to study the brain. And so another important factor in all this while I was working hard is that I applied to a program called Career Opportunities in Research. So today, if you go to, say, like UC Irvine or to CSU Fullerton or Cal State Fullerton, I think they have a program called MARC. And MARC, the idea here, that what MARC stands for is maximizing access to research careers. And so this is all sponsored by the National Institutes of Health. And so I met Dr. John Jung, who was a professor there, and he really recommended that I applied to this program. And so I got into the program, I quit my job serving beer and pizza on campus, and I was awarded a scholarship, and I earned a stipend on top of that. My mind was completely blown away that this opportunity existed. It allowed me to focus for the first time just on my study. And so it was just an amazing blessing to have. And I'm very grateful to the university, Cal State Long Beach, but also Dr. John Jump. So that again allowed me to actually pay for my rent, go to school full time. The other part of this is that the only thing I had to do was continue to excel in school and my classes, but I also had to do research in a laboratory. So I spent a little time in one laboratory at CSU Long Beach. But then as summer came around, part of the program was to send these scholars, including myself, to a laboratory. And I was, again, smitten by the idea of doing neuroscience. And so I was fortunate to have the right people around me, the people that would be supportive. And I got a chance to interview at a research laboratory in neuroscience at the University of Southern California, studying learning and memory. And so I was chomping at the bit. This was exciting. This was like the most exciting thing ever. And so I worked there all summer. Given that it's quite far away, University of Southern California from Long Beach, I drove in during the weekends and during break, and I spent the whole weekend there working with a graduate student in the lab. And so that really, I just was happy to be there, being able to, do some amazing work. We were recording from the brain as learning was happening in an animal. And so we were measuring the activity of the brain as learning was happening. And so just the ability to try to study how the brain learns was just, again, it just left me in awe and really thinking about what I would like to do in the future. So I absolutely love my research, as I've already mentioned. I got to work with this grad student named Dave King, as I mentioned. I had mentioned his name. And with the advocacy of him, but also with the advocacy of one of the most eminent neuroscientists at USC, professor named Richard F. Thompson. My hard work earned me eventually admissions into the University of Southern California, Behavioral Neuroscience PhD program. And I have to say that getting into this program was probably my greatest academic achievement. For that reason that I was on academic probation, I had to retake classes, I had to work hard, I could only get A's, right, to get my GPA up. And so I was excited. I was just given the opportunity. And while the first year of graduate school was really, really hard, it was the most amazing time in my life to be able to do research, take classes, and TA for other classes. Again, it was a very productive and exciting time. I will never forget that. It laid this important foundation of learning to become a scientist, asking the right questions, and being mentored by really amazing people. All right. So the hardest part was the first year for me. Everything else was pretty good. I graduated with my PhD, and I continued my research at UCLA with a leading expert in the study in the world, one of the leading experts in the world to study fear. His name was Michael Fanslow. So I spent the next 6 years really grinding at the bench, trying to publish really excellent, publishing excellent work in journals. And then I started developing my own research idea questions, things that I wanted to study in my own laboratory. During that time at UCLA, I met my wife. Actually, my wife wasn't at UCLA, but she worked at UC Santa Barbara. And then in that process, we got married, and then I received the job offer to open my own laboratory and teach at the university at Albany. So my wife, who was a counseling psychologist, and we left Southern California, and we moved all the way across the country to upstate New York. So, if you love thinking and doing science with a little guidance and hard work, it can take you pretty far. For us, it took us 3,000 miles across the country. And so this is where we're at right now at the University of Albany. It's part of the State University of New York system. So as I mentioned, we're in the capital city of New York. We're situated near the Adirondack Mountains and Catskills. We're a few hours from Montreal, the Canadian border. So there's a lot here. However, I will say it's pretty cold. In fact, we had snow this morning. So again, things that I had to get used to. And then the picture at the top is my lab, our building that is the life sciences building, where some of the psychologists in our department are housed with research laboratories. So I'm very fortunate to have this opportunity. I have a number of graduate students, and I'll talk a little bit about their work, and I'll acknowledge them at the end as well. Right. So what do we study? Well, we study fear. And well, what is a good definition of fear? So give me a second. I'll let you read the Yoda quote. Well, according to Yoda, fear is the path to the dark side. Fear leads to anger, and then anger leads to hate, and then hate leads to suffering. Very philosophical. But if you're to study a phenomenon such as fear, you really need to have a very good definition, right? You need an operational definition. And so the definition that I sort of, in some ways, inherited from my postdoc mentor's lab of fear, and I use the same definition to this point, to this date, which is the perception and recognition of danger, the learning and remembering of dangerous experiences, and therefore the coordination of defensive behaviors to environmental threats. So what I'm getting at here is a couple of things. One is our perception and recognition of danger actually requires largely the ability to recognize and learn what are things that are dangerous in our environment, right? And so when we experience something that's considered dangerous, we learn about those dangers. What things in our environment lead to those dangers, right? And what this allows us to do is coordinate appropriate behaviors to these threats that are in our environment. We call these defensive behaviors. Now, fear itself is normal, right? Without exhibiting fear, we probably wouldn't survive very long on this earth, right? Now, all of us probably think, well, when I'm afraid of something, how do I respond, right? Well, the reaction to potential threats can come in many different forms. The one thing that we might note is that our heart rate might increase, right? We also might show increases in sweating, right? Our pupils might dilate, as my daughter has pointed out. I don't know where she's learning this, but she's only 7 right now. Our blood pressure can change, right? Our breathing changes. We become more vigilant to the things that are around us, right? We might become more prone to startle, right? If all of a sudden you're exhibiting sort of certain, you're in a fear state, if somebody comes from behind you and taps you on the shoulder, you might jump, right? Others, you can exhibit a facial expression related to fear. We can also measure certain hormones, such as stress hormones. These become elevated. Now, one of the things that ties all of these things together is there are certain behaviors that are exhibited not just in people, but also in different other species of animals, which is when mammals are exposed to threats, they will show 3 types of behaviors generally. One is they will either freeze, remain motionless when they detect threat, depending on the severity of the threat or its location, right? They will remain motionless. So you might ask, well, why is that being motionless actually helpful? That doesn't mean that you might get hurt. Well, when we think about the natural relationship between a predator and prey, right? Say a lion, right? Hunting a gazelle or something to that effect, right? In some ways, freezing itself is adaptive because a predator actually responds to movement, right? And so remaining motionless, like, a squirrel, right, might freeze, for example, when you're walking your dog. It actually minimizes potential producing that chase response. Now, sometimes the most appropriate response is to run or flee, right? And animals will exhibit that depending on how proximal or how close the threat is. And sometimes the threat may be too close, and the only thing left to do is to fight. Okay. So these are, I think, commonly shared across different animals, not just in humans, or in rats and mice, which I'll be focusing mostly. Okay. So in our laboratory, we're primarily focusing on using rats to study fear. Okay, so imagine here is a rat. I'm just going to give you an example potentially that might actually happen. Even though this rat doesn't normally live in the wild. This is a domesticated rat here you're looking at. Let's say this rat is exploring this rock, and guess what happens? A snake pops up, right? And it attacks the rat. Well, the first thing that the rat will probably do is probably flee if it's being attacked, okay? And so it will either run or freeze. In this case, being attacked by a snake, right, it's already making this attack movement is to run or flee. Now, let's say that this rat decides it needs to get next to that rock for some reason. Maybe there's food that it hid somewhere, or there's a source of food, and it's willing to take the chance, right, to go past this rock again. Well, it might approach it by moving really slowly and then freezing, and then attending to sounds as well as visual aspects that might relate to the presence of the snake, right? So it's really important that the rat display a change in its behavior that allows it to tend to its environment and minimize movement and therefore attack. A snake again moves, will attack based on movements of a prey. Now, what I just try to point out here is a relationship that the rat here is learning about the relationship between that rock and that snake, because that snake perhaps resides under that rock. Whenever it was around that rock, that snake popped out, right? And so it's gaining a knowledge about the relationship. And so in some ways, this is a association, right? That rock, it becomes associated with the snake, right? And in doing so, that rat now has to display appropriate behavior under these conditions of threat. Okay. Now, this might sound very familiar to you if you think about Pavlovian conditioning. You guys have probably learned a little bit about Pavlov and classical conditioning. So my lab is interested in studying fear and the brain correlates of fear, okay, very broadly. All right. So how do we study this actually in the laboratory? I don't have snakes in my lab. I don't have rocks in my lab either. So what we do is our main subject, even though we use mice as well, are rats. And what we do is put them in what we call a Skinner box or an instrumental box that looks like this. Okay. And so the rat is put into the box and it's allowed to explore the box. Okay. So the first time the rat is in the box, it's learning about the environment which it's in. Okay. And then all of a sudden we give a mild foot shock. So see the grid floor that's in the image? We deliver a shock. So a little bit of current that causes a mild and momentary sense of pain in the paws, and that goes away really quickly. Okay. And so basically the rat is learning that anytime this rat is in this box, that there's potentially something threatening in its environment, right? In this case, it's the environment of being in that box becoming associated with shock. Okay. Now, if we so the idea here is that the context or this acts as the conditioned stimulus. Remember Pavlov, right? Where Pavlov's experiments were involved pairing the ringing of a bell with food. These experiments are pairing an environment, a box, with a foot shock, right? In Pavlov's experiments, food, a bell, and food. This is our unconditioned stimulus. Okay. Now, if we bring our rat back, say 24 hours later, and we put him back in this box, and we're not going to shock him. We're not going to give a foot shock or anything. We're just going to put him in the box. And what this rat will do, upon being placed in it, it will start exhibiting a freezing response. Okay. It will lie still, even though this is not a video. The rat will sit there and display no movement at all. It'll go through periods of time. And so 90% of the time the rat is in this box, for example, or 80% of the time, it will freeze. It won't be exploring the box anymore. It had learned the association between the box and the shock, the foot shock that they got. And so in this case, the conditioned response is to freeze. Okay. Again, consistent with how how Pavlov described his experiments, we're describing it within that classical conditioning perspective. Okay. So here, fear is learned to the box, right? And how we measure fear is by measuring the freezing in these subjects. What percentage of the time do they freeze? That gives us an idea of how much fear there is. All right. Now, if that is how we study this, the behavior of the animals, right? These are the conditions that produce fear. The three main areas of the brain. So you're looking here as a drawing of the rat brain. And the three areas of the brain that I want you to focus on are, one, the amygdala. So the amygdala is part of the brain that's involved in emotion processing. So how we process emotion. And this has largely been demonstrated not just in people, but also rats and mice and in monkeys as well, that the amygdala is important for fear. Another brain region I want you to focus on here is the hippocampus. So the hippocampus is a part of the brain that processes the environment. It learns about where you are in a given environment in the world. So, for example, I'm in my office. My hippocampus knows about my office. I'm here every single day, maybe except Saturday. And so my hippocampus has a strong representation. It encodes information about its environment, right? Some days my office, I might be, it might be a tough day and I don't want to go to work. Maybe my amygdala is telling me that it's not a good day to go to work. All right. The third different brain region is the prefrontal cortex here in green. So the prefrontal cortex is important because it's generally described to be involved in decision-making, and so it allows us, in some ways, it has a direct influence in controlling the amygdala, but also it's involved in communicating with the hippocampus. So the prefrontal cortex that sits, like its name suggests, in the frontal part of the cortex of the brain. So in the fear conditioning, the fear experiments that I described, the hippocampus is involved in coding the environment in which the rat is. The rat is learning about the environment, and this is being learned by the hippocampus. However, let's say that we introduce a foot shock. Well, that foot shock is going to cause the amygdala to become activated. Okay. Now, the other important thing to understand is that the amygdala and hippocampus are connected to each other. So the cells in the hippocampus, some of the cells can exit and reach the amygdala. And some of the cells in the amygdala, the neurons, they can send their long axons to form synapses onto neurons in the hippocampus. So they can communicate in both directions. Then we have the prefrontal cortex. The prefrontal cortex also can connect to the amygdala. Okay. And so information can go from the amygdala to the prefrontal cortex as well. So bidirectionality. Okay. And then the hippocampus and prefrontal cortex are also connected with each other as well. Okay. And so eventually, if the amygdala is involved in encoding, so let's start over here. So again, as I mentioned here, the hippocampus encodes information about the environment, right? And the shock is encoded by the amygdala. Now, the hippocampus also communicates to the amygdala. And the idea is that the relationship between this box, the environment, and the shock will become associated in the amygdala. And so just returning the animal back to the environment in which it was previously shocked will cause the hippocampus as well as the prefrontal cortex to activate the amygdala and then generate the freezing response in our subjects. Or in humans, the sense of fear and dread. Okay. So, whoops. Pardon me, I'm going to move some of these things around a little bit here. So my lab actually is focused on really three different aspects, three things about fear that are, I think, particularly interesting. One is, how does fear change depending on our developmental age, right? Everything that I've talked about is about fear in the adult animal. But as we all know that fear, we also can learn fear when we're younger as juveniles, before, just around that period right before we go through puberty, but also during infancy, okay? And so one of the questions we're interested in is, how do the connections between the prefrontal cortex and the amygdala change across these different ages? And how does the hippocampus and amygdala connections change also? So that's one thing that we're interested in, sort of the development of fear over long periods of time. A second question we're interested in, and it's one that's also related to related to development, are sex differences in fear. Okay, and so we can study this both in male and female rats, and then we can look at how the amygdala here and how the hippocampus and the prefrontal cortex, which you don't see here, how are they activated? Are they activated the same way, males and females, right, by the same experience, or are they activated differently? Okay. Moreover, because when we say sex differences in fear, it doesn't mean just focusing on the adults. We can actually ask the same question in the juvenile animals. So that's the second part of what we do. And the last part of what we do is also, I think, particularly interesting. And I think the first two things that I described are about normal fear, right? Fear is normal, it's regulated by depending on how much threat there is in the environment, right? That we exhibit the right amount of fear so that we could survive. Now, what happens when the threat becomes very strong or traumatic? And so we're interested in how experiences that might be considered traumatic, how they impact later on fear. In fact, we're actually asking this question not 24 hours after a trauma, but almost 2 months later. So rats, for example, their lifespan, they live probably, no more than three years. And so their developmental time windows are very different. So we might ask, well, if the animal was exposed to a traumatic experience, let's say as a juvenile, what happens to their ability to learn about fear later on as an adult, months later, right? And so our lab is focused on studying this in different brain regions as well. What are the changes that happen, right? And so part of this is, of course, that this work has some relevance because it relates to or stress and trauma related disorders like post-traumatic stress disorder, which is a debilitating mental health problem in our country. And particularly, it's also more prevalent in females than males. So we're going to ask questions about the age in which trauma occurs and the sex of the animal. Does that matter in how it impacts, right, trauma and its effects later on in fear, right? So one, and I'm not going to get too much into the details here, is that one of the things that in order to establish how one area of the brain is connected to another, you have to go into the brain and inject these, what we call tracers that go into the brain. And so these are injected in specific parts of the brain, such as the amygdala. And these tracers will actually be taken up by the cells, and then they will travel to the origin of where the connection of another set of brain regions connects to that part of the brain. And so it allows us to look at the amygdala, but also label the brain, such as the prefrontal cortex on here on the left, as well as look at how, so again, prefrontal cortex is connected to the amygdala, which is labeled here DLA, and then how the hippocampus is connected to the BLA or the amygdala itself. Sorry for all these acronyms. There's a lot of terms. I don't want to go down that road right now. So this is work done by one of my first PhD students, Anthony Santarelli. And so he was interested in this question of looking at the connections between prefrontal cortex and amygdala at different age points in infancy. So in a rat, infancy is between birth and about 19 days of age. In juvenile age, it's about 21 and just before puberty. And then adolescence is thought to be when puberty occurs, and then adulthood is about 75 days of age. So all of these regions make up the prefrontal cortex. So note here in the black line, this is the number of cells in the prefrontal cortex that project to the amygdala. What's particularly interesting is that the prefrontal cortex seems to be most connected to the amygdala during this juvenile period. And then it declines during adolescence and adulthood. There is one exception to this. It's a very specific portion of the prefrontal cortex that actually shows growth across these time windows. And if my daughter pops up behind me, because I had to pick her up, I apologize. And so she might pop up and say hello behind me. Anthony then looked at the connections between the hippocampus and the amygdala as well. So on the right panel here are the number of neurons in the hippocampus that connect to the amygdala across these age points. So what you see here, unlike the prefrontal cortex, is that there's continued growth of connections through adulthood. Okay, so this is a very different pattern we see, right? Prefrontal cortex here in green is most strongly connected during this juvenile period. The connections are actually somewhat lost or pruned during adolescence, whereas for the hippocampus, growth happens. Okay, so that really sort of gets us at this point, suggests that, oh, these connections change their dynamic, they develop. It might suggest something unique about this juvenile period. Okay, now the one thing about this is that these experiments were all performed in male animals, male rats. So we didn't specifically study here in females. Now, part of that led us to the next series of experiments to examine sex differences as they emerge from the juvenile to adult period. Okay. And what we're going to do is measure the activation of these three brain regions at in juvenile and adults, male and female animals. All right. So this work was performed by a graduate student, Loriana Colon. Excuse me, Dr. Loriana Colon. And so what she did here was to examine how context fear conditioning affects the activation of each of these brain regions in our rats. Okay. The first thing is that what you're looking at on the right here is the animal's behavior. So we're looking on this vertical axis here is the percent freezing, the percent time the animal is freezing to a box into the context in which it was previously shocked. We call this context fear retrieval because they're retrieving the fear of that context, the box. Notably here, what is really interesting is that in males, fear conditioning or freezing is rather low in juveniles and then increases in adulthood. In females, it's the opposite pattern. Freezing is highest in our female juvenile rats and then decreases during adulthood. So we see this really interesting pattern. Now, this is the, whoops, sorry, guys. The sex difference has been all before noted, but what we're arguing here, what we're demonstrating here is that this difference actually emerges uniquely, apologies again, and has a developmental pattern. Okay. So I'm not going to show you all of the data from this, but what we find is that if we measure the activation of the brain, the prefrontal cortex, we find that there's no difference in males and females in the activation of the brain when animals are exhibiting fear to the context. However, juveniles show greater amounts of activation than adults, irregardless of whether they're male or female rats. Remember I told you about the connections being peaking, the highest level of connections between prefrontal cortex happens during the juvenile period? Well, there you go. This is the activation of the brain is greater. The prefrontal cortex is greater at this time. The amygdala actually shows equal amounts of activation, whether the animal's juvenile or adult, or male or female. What about the hippocampus? Well, in juveniles, females show greater hippocampal activation than males. In adults, males show greater hippocampal activation than females. The pattern that we see with behavior corresponds very well with what's happening in the hippocampus. Okay. So the last set of experiments, one experiment I'm going to describe is looking at how a traumatic experience in these rats during the juvenile or adult period impacts the animal's ability to learn about fear later on. Okay. And so this is work by one of my graduate students right now, Samantha Hetherington. And so she's looking at juvenile and adult rats, female and male. And what she's doing is exposing these rats to either no stress, so one group has no stress, so it's a control group, right? One group has low stress, another has moderate, and another has high stress. And then 2 months later, they're brought into a novel box and they're just given a single shock. Okay. If we look at the females first, what we find here is that no matter whether it's low, moderate, or high, fear conditioning is enhanced 2 months later in the juveniles. Okay. In the adult animals that are given this stress condition, they're basically only group that showed higher levels of percent freezing or fear were the adults that were given high levels of stress. So low and moderate was not sufficient, being low and moderate condition was not sufficient to produce any change. All right, now in males, the pattern is almost completely opposite. If we're looking at males in the juvenile age, if they're given stress and we wait 2 months later, overall, we don't see an effect statistically of the stressor. So it doesn't produce a long-lasting effect. But if the animals were adults, the males were adults, and then we wait 2 months, we see what we get an effect where high produces the highest amount of fear learning, this point, and moderate shows lower, and then low has the lowest relative to the control, but they're all elevated. So the stress is actually enhancing fear learning, and it lasts for a couple of months. So this itself is pretty compelling, and we're looking at right now the activation of the prefrontal cortex. So we're studying a lot of really interesting things I don't have time to get into, but I think overall what our work has demonstrated so far is that the connections between within the regions of the brain that are involved in fear and decision making, as well as learning about the environment, continue to develop over the course, right, of infancy, during the juvenile period through adulthood. We're also pointing out here that there are sex differences. In fact, the sex differences seem to develop uniquely in males and females when it comes to fear and the activation of these same brain regions. And then ultimately, how is this clinically relevant? We think that if these are examples of normal fear and how it's processed by young and adults, male and females, these studies are getting at how fear can change depending on how much trauma you experience, right? Now, obviously, we're talking about rats, and so we can't really conclude too much about humans. But I think it starts beginning telling an important story about the timing of traumatic experiences, whether they occur in early life versus adult life, and how they might be uniquely affected. So I want to thank all of the students that have done the work. I'm actually missing a couple of students that have not been added here, graduate students and undergrads in my laboratory. And I have a couple of high school students. I also want to thank my collaborators. Some of these names might look familiar to Melissa. And I also want to thank my mentors. These are really important people because I wouldn't be here today without them. Many of them have actually passed on. And so whether it's at Pasadena City College, Dr. Gene Volkman has meant the world to me and helped me get here. John Jung, as I mentioned, from CSU Long Beach, and then Richard Thompson at USC, and then my other mentor, Michael Fanslow. So, and then I also have to thank where the funding comes from, which is the National Institutes of Health and the state of New York. And I also want to thank you all for listening to me. I don't know how many of you are still here, but I'm happy to answer questions if you have any questions.
Thank you.
Okay.
I have a few people leave for class, I think, but does anyone have any? They have to go to class occasionally.
No worries. Thank you.
Any questions? Were there any questions? I know Emily asked for people to submit some in advance, possibly. Were there any questions or?
I had,
Okay, sweet question. I mean, it doesn't just have to be about the research, anything about like I thought it was really interesting, and thank you for sharing the pathway to getting there, because lots of connections with first generation students, community college. You know, I think that definitely speaks to our audience.
Well, I'm glad that I could speak to it because this is my experience as well, and like I said, it's an honor to be able to talk to you guys. Hopefully you guys can see a path moving forward. Having the opportunity, you know, I think each day you guys have an opportunity to move in whatever direction you want to move into, whether it's being a scientist or not, or whatever you want to do in life. And I think that's for me the most important thing.
Yeah, we have 3 already submitted prepared questions for you.
Sure.
One being, what advice do you have for someone interested in going to graduate school for psychology or neuroscience?
Oh, well, I think part of my advice is learning about what graduate school is about. And because I can't offer advice and without telling you what it really is. And so it's sort of this interesting mix. So you guys are taking classes, right? And you're learning a lot from your classes. Well, being a graduate student is very unique because not only are you taking classes, but you're also doing research that creates the knowledge that you're learning, right? And so it's mind-blowing when you put it together. But moreover, then your job is you also have to be a teaching assistant. So you're teaching other students while you're learning and you're doing research. So it's a very different thing from just taking classes. I think it's just amazing because even today, to this point, when I get a chance to teach, I feel like it actually helps my research more because it makes me think and explain things in a way that simplifies them. And really about science is about simplifying ideas, right, into manageable hypotheses. And so my advice, of course, is if I would have any advice is to one is that graduate school, now I'm talking about a PhD program, is not 2 years, 1 year. It's perhaps not even 5 years. It can be 5 to 7 years of work. But if you love what you get a chance to do in graduate school, it doesn't really matter. Hopefully you get surrounded by good mentors. So you really want to love what you do. And it's like, I also mentioned that the other part of this is that you're training to become a professional, right? And you get to train with someone that already is there, right? To be a professional, it takes many, many hours of training to learn, right? So imagine somebody's an Olympic skater, right? How many hours do they put into it? In some ways, being a scientist takes that same amount of training, right? And it's important to put in the effort. And so hopefully that comes through, and it helps you become, what you want to become as a scientist, whether you want to become a teacher or you want, or you want to be a bench scientist or run your own laboratory. So I think the other thing I want to point out, in many PhD programs in graduate school, is that actually you don't pay tuition. There are exceptions depending on whether I think you're going into clinical programs, but in the sciences, within psychological sciences, you get actually your tuition covered as a graduate student if you get into the program. And you get a stipend. Now, stipend may not be the greatest amount of money, but it is enough to survive, barely survive, meaning that you're not going to be very rich while you're a graduate student. And I'll say maybe that's an understatement, but it affords many opportunities. So you just have to, again, picking your advisor is another important thing. If you have a real strong interest in something in psychology and you can find someone that shares that same interest in research, that's a good relationship. You're establishing a relationship with your mentor, and that's really important. So I don't know, that would be some of my advice. I can go on for hours about this because this is what I do. I talk to grad students, I talk to undergrads about going to graduate school. I'm actually one of the mentors for the MARC program here at our university as well. So
Nice, thank you. That was really insightful. Just to get to the other question, what are some challenges and rewards of being a professor in your field?
let's see, rewards are generally be able to teach and to communicate what you love to do, what you're interested in, right? For me, I'm fascinated by the brain, and I get to talk to students about this. They put me on a stage and I talk to 200 students or 20 students or even 4 students. And that's an awesome experience to be able to share that, right? And that's really rewarding. Doing science to me is what got me interested. Is like actually doing the research. It's like being in a laboratory is like putting me, watch Disney cartoons, and all of a sudden now you put me in Disneyland, right? You get all the tools, everything is there, and now you get to do the science. So yeah, I would say those are the pluses. Some of the, I say, limitations and cons, I would say that it takes a lot of work and focus. While you're a graduate student, sometimes you don't have the full experience of other people, your friends around you, because you are working so much and you love it. And then as a professor, I think we work long hours, we write a lot, and so we're writing grants, and so it can be exhausting. But I prioritize certain things, especially family. And so I think as long as you can prioritize and have some control of that, it's important. And once you become a professor and you get tenure, you have great job security on top of that. So that's another plus, I would say.
So I think we have another question. I'm just gonna preface it. My computer is making strange noises, which normally it does that right before it decides the battery is going to die. It says it's at 19%, but you never know with this thing. So I apologize if it just cuts out on you. Well, here, next one, it will cut out on everybody at the same time.
No
I think
worries.
one more question, right?
Yeah, just one last question. What does a typical day look like for you as a professor and researcher?
Here's where it gets interesting. Every day it seems to be different. I have so many things going on that okay, for example, let's do a week. Okay, Tuesdays, let's see, Wednesdays I teach, and I teach Fridays. So I have like 2 classes on Friday, and I have 1 long class on Wednesday, which is basically 2 classes. But then I basically teach and I have discussions with graduate students. On some days I have lab meetings where I get all of my graduate students and undergrads, and we have a meeting and talk about science and what they're working on. Other days I meet with them individually, talk about their research. And then I have lots of meetings with the dean of the college, with my colleagues. I don't do research anymore. I don't actually go into the lab and work with pipettes or brains anymore. My graduate students do all of that at this point, even though I come in and say hello all the time. My job is so diverse, whether it's involved in outreach, the MARC program, trying to get students to go into PhD programs. It's really important. I look at admissions to PhD programs. So we decide which students get into our program. We only take a few students every year. So I love to exercise still because it keeps my sanity. So our job is sedentary. That's a con. That's a bad thing about it, because I sit in my chair a lot. But exercise and doing things that's important to keeping you healthy is, I think, is important, like eating healthy and exercising. So I don't know if that answered your questions, or did I just completely go off? And maybe my brain is deprived of nutrients at this point as well.
No, yeah, it did. Thank you so much. I just want to extend our gratitude and say we're really grateful for you taking the time out of your day to join us, even when you're on a different coast. And I think that this guest speaking event was really helpful. But yeah, do you guys have any other questions, or should we wrap up things?
All right, well, thank you so much. It was good to see you on Zoom. I know on Facebook every once in a while, but
Thank you, Melissa. I'm glad to see you as well.
yeah, it was nice.
Thank you, guys. Thank you, everyone.
Thank you.
Thank you so much.
Wonderful day.
You too.
You too.
Have a good evening.
Oh.
Bye.
Go get some food.
Yeah.
Bye, Melissa.
Bye. All right. So now.